And Dr. Andrew Benson is next, and he's going to be discussing prospects for diagnosis by sequencing. Dr. Benson received his PhD in microbiology from the University of Texas Health Science Center at San Antonio and completed postdoctoral work in molecular biology at Princeton University. He currently holds the W.W. Marshall Distinguished Professorship in Biotechnology at the University of Nebraska-Lincoln. His research focuses on the interaction between host genetics, environment, and gastrointestinal microbiota, as well as genome evolution in pathogenic bacteria and the evolution and development of gut microflora. Dr. Benson?
I think one of these times I'm going to put something really interesting in one of those bios, you know, like I was arrested for something, just to see if anybody noticed. So many of you are probably wondering, A, why I'm here, and B, why I'm talking. Should I move this down a little bit? Yeah, a little high. B, why I'm talking about this particular subject. And I'm here because I got suckered in. I mean, Jim Reese talked me into sequencing a couple of nasal pharyngeal swab samples several years ago, and also into writing a grant with him. And then B, we do have a project that relates to sugar toxin producing E. coli or STECs, where I think we can glean a lot of information about the prospects for using sequencing to do diagnosis. And so I'm going to tell you a couple of stories today that are all part of one major project that we have going on. And they're sort of two little vignettes, but they're all part of one big picture. So let me tell you first a little bit about STECs. I apologize, I probably won't talk much about BRD for the rest of the entire talk, but you can translate this, as you'll see as we go through this, quite easily to this polymicrobial disease of BRD. So STEC and the bovine gut microbiome, we've heard a bit about the STECs already. I just want to talk a little bit about the distribution that you see of these things in the animals. So STEC prevalence can be quite high at any given time. The shedding patterns can vary significantly even within a single animal. And remember, the shedding is the problem. It doesn't cause any overt symptoms in the animals, but obviously causes problems in humans if it's the wrong STEC. Shedding patterns will show a log normal distribution across the population of animals. That's important because if we think of it as a complex trait and a quantitative trait. And any given STEC genotype can be transient or it can be long-term. So it may be there for some time or it can be very transient. And this comes back to a lot of the strain-to-strain variation. These guys are the kings of genome variation, it turns out, at least among what we found around in a lot of the pathogens. And at least one niche for at least 0157H7 STEC uses exactly the same attachments machinery in the host, that is the bovine, as it does in the human. So that's an important aspect to the ecology. But really, to understand the ecology of this whole system, we really need to understand how this gut colonic ecosystem assembles and then how it functions. And so that's really what's driving our research. And I'm not going to talk too much about that other than how we're generating some of that data. I'm going to try and instead lead you where some of that data can be applied. What got us started, really headed down the road, was the fact that most of the studies of STEC shedding, STEC colonization have all focused on management and environmental factors. And virtually nothing was known about the role of bovine genetic diversity. And so that's really what got us intrigued, especially knowing with some of the rich databases and some of the rich genetic resources that were going to be available within the bovine. That got us intrigued into trying to get down this path. And we started down this path early on because we noted that in mouse intercrosses, the gut microbiome actually behaves as a complex polygenic trait. And so this was an experiment that I did with Daniel Pomp several years ago where we looked in a mouse intercross population and we phenotyped the gut microbiota and treated that as a trait, actually as a collection of traits. And a collection of several traits. And so if you do QTL mapping in that type of experiment, you genotype the animals and look for the association with the variation in the traits, what you find is that there are several loci in the bovine, or in the mouse genome here on the outer circle that control the relative abundance of taxa inside the GI tract of those same mice. And so anywhere you see a little red line here, that's a QTL. And inside of the diagram, that's a phylogram. And it's sort of an average phylogram across all 700 animals in the population we studied. And they're color-coded by their phylogenetic relationship. And what you'll notice if you look at the color codes up here of the confidence intervals, the QTL, what you'll find is that the genetic control is actually dispersed across the phylogenetic space that's recognized or that's occupied by the bacteria. So it's not as if host genetics is focusing in on one given group of organisms. It's focusing on several organisms across the phylogenetic space. So you'll see members of the firmicutes that are in the blues. You'll see members of the Bacteroides that are in the reds distributed around that chromosome. And there are also several loci where there are more than one taxa. So there are several pleiotropic loci. So it's one gene or one locus actually controlling whole groups of organisms, not just one particular organism. So that was what we learned early on using taxa, using the species of bacteria as individual traits or as collective traits. And the last two slides I'll show you with mice, we're now using their functions, because really it's probably the functions that the host actually cares about. It probably doesn't care necessarily which organism is contributing as long as the function is there. And so we're doing whole metagenome sequencing on these samples now. And this represents the whole metagenome and the metabolic content, sort of the metabolic universe, so to speak, of the microbiome from a population, another different population of animals that we've been studying. So here's the microbiome here, and I'm going to superimpose on that the pathways that are influenced by host genetics. And there's quite a number of them, and I just wanted to point a couple of them out here. Glutamate metabolism is sitting right down there, and you just heard Mark Light talk a lot about this interaction of the gut-brain axis. This may be another example of it showing up here. We also have butyrate production over here on this side that's heavily controlled by the host. Again, this is the butyrate is one of it's probably the molecule, the currency of energy down low in the GI tract. So this starts to make sense when you start looking at the interaction between bacterium and host. So now let's take that's in mice. Now let's take that and translate that back into the Shiga toxin producing E. coli story. Sorry for the little diversion in mice, but that's to show you the tools. So here's the way we're looking at this. So STEC shedding will show a log normal distribution. So this is a log 10 scale down here on the bottom, distribution across the population of animals, and of course there will always be a lot of animals that are not shedding. And the question is this actually a complex trait? And is it controlled by components of the colonic microbiome where the organism likes to reside and host genetics? So that's the whole idea going into this study. And in order to get at that, I should say, as an outcome that's going to be relevant to the topic here, at least two of the different types of sequencing we're doing are very relevant to, A, the colonic microbiome, which would represent a polygenic situation, a polymicrobial situation. And also, we're trying some diagnostic sequencing directly on enrichment cultures. So I'm going to talk about those two things as we get down the road here into the talk. So the study design, just so you know what we're talking about within this study, so there's 1,500 animals. These are animals sourced out of the U.S. mark herd. They represent the 16 most common beef cattle breeds. At least 4 finishing diets. We have one final cohort that's being pushed through this year. And I think we have 2 geographic locations that we ended up with. But again, all the animals were sourced out of the mark herd. Each animal is being genotyped, and the ratio of 50 Ks to 70 Ks changes depending on the price daily. So originally that was the ratio. And the phenotyping, grab samples from the rectum. The EHEX and the STEC still have to be done by just hard grind it and find it plating-based microbiology. It's a real pain in the butt, and the guys out at US Mark do that, and I give them all the praise in the world for it because that is the hardest part of this whole project. That and collecting the samples. They actually take the fecal samples, composite them because they're sampling the animal 7 time points for each animal, composite all 7, put the composite in a 96-well plate and send it to us. That's pretty nice. I give the guys out there just tons of praise for doing that part of it, and they never complain about it. We're doing now whole metagenome sequencing, and you can see I crossed that out because originally we were going to do 16S. The sequencing technology has changed. It's changing rapidly. It's a very fast-moving train. Virtually any monkey can get something sequenced now. And I'll get on my soapbox here and say the worst thing that can walk in your door if you're a data scientist is a guy with 5 samples and $20,000, because they'll waste more of your time than you can imagine. So the data science, somebody knows exactly what I'm talking about. The data science is huge in this because there's a massive amount of data. You've got to collect it and process it properly in order to get the information out that you want. Okay, I'll get off that soapbox. So this is just to highlight the team here. This is my team. I'll come back and talk about them later. And again, the guys out at US, Mark, Wells, and Bono, who are doing all of the microbiology. Min-Sok Kim, who's done some of the early data analysis, and Larry Kuhn and Warren Snelling, who are doing the host genetics side. To date, we've sampled 1,219 animals, so now we're sufficiently powered to go ahead and do a preliminary association study. We've done shotgun metagenome sequencing on all 1,200 of these animals. That's a lot of data. 1 to 20 million paired-in Illumina reads from each animal. So that's the amount of data that we're dealing with across this population. So now the question becomes, we're generating all this data. How do we turn it into something that's quantitative that we can actually use? And it's full metagenome data because I wanted function in addition to just the taxa. So somehow we've got to come up with a way with quantifying the taxa that are there and quantifying the functions that are there. And so what we do, and we have very few, unlike the Human Genome Microbiome Project, we have very few reference whole genome sequences that we can map things onto. So we have to generate our reference on the fly. And that's how we've been doing it, and it seems to work quite well. And so the strategy is, so here's our metagenome community up here, and each of our little circles is a different species of bacteria. And the sequencing process, essentially, you can just think of these each as a different book. And if the two bacteria are very closely related, it might be 2 versions of the same book, right? For edition 1, edition 2. If they're not very closely related, they could be 2 very different books. And think of the book now not as a collection of words, but as just one big word. Take all the spaces out. Right? That's essentially what you're looking at in a metagenomic population. You now break that book up multiple times, each one multiple times, into little pieces, and you get your reads. That's just the letters on the very ends of your pieces. And that's your starting material. That's what you're getting from the sequencing process. And somehow now, you've got to try and reassemble this back into the books and quantify how many of each book that you had. So it becomes a linguistic, it becomes an algorithmic problem very quickly. And so the quickest way to get through that is to, before you try and assemble anything, is try and sort all these little pieces here into blocks that probably came from the same book. Right? That's the best you can do. And you do that by looking at the short words that are present in them. And those are called kamers. And so the first thing we do is sort those into these blocks that probably represent maybe a genus. And then you assemble back into the books. Because if you try and do it without sorting them first, you'll get a jumbled mess. So this sort of helps get by some of that jumbling process. And we get chunks of DNA now from our population, contigs and scaffolds that are linked together by these reads. And the next thing we do is to take all of them. So here we call each of those a contig. Here's our population we started with. We had 1,152 samples that we got good sequence from. We had 3,200 contigs that were over 10 kb. Anything smaller than that we threw out. We considered was not very useful. Next thing you have to do is try and figure out who the heck these things came from, because you have no idea what organism they came from. So those are pooled together using a couple of different strategies. And this represents then what we call our genus level or contig bins or our reference assembly. So this is our assembly of taxa based solely on the metagenomic information, solely on the DNA that we extracted out of this fecal material. So the next step then is to quantify those. So we take our reference assembly, shown right here, and we map all the reads from animal 1 across that reference assembly, and we essentially count how many times did we get reads that mapped onto taxa 1, 2, and 3. Here's animal 1, here's animal 2. You get the idea. That's how we quantify that. We quantify gene function a little bit differently. So for gene function, we annotate all of those contigs. We find all the genes in there. And now we do the same thing. We map our reads onto them. And now I don't care which taxon they came from, but if I have a gene that or a read that maps to gene A from animal 1 and animal 2, they both get a count for it. So now I'm mapping function, or I'm counting function independent of the organisms from which it originated. So I can look at taxa and function totally independently in this project. So just to give you an idea for what that looks like, this is a distribution of the number of total reads that we got across all those animals. We assembled each of those animals individually into the full reference assembly. And I'm going to skip that and go to this. This is box and whisker plots now. And this is a log scale down here on the bottom, starting with most abundant here to least abundant down this direction. And so these are about 150. You're not seeing every one of them, but these are about 150 of the top organisms that are present. They're the most abundant organisms that are present across 75% of our animals. And the important thing I wanted to point out here about using full metagenome sequencing is we don't have the biases that are inherent to 16S sequencing, meaning we can see everything that's there, or essentially everything that's there. And so we're picking up all the methanogens now that we never would have seen before because our 16S DNA doesn't get amplified. They're members of the archaebacteria. So now we can actually see those in our populations. So now the question becomes, can I now connect these taxonomic and these functional abundances with anything that's in the microbiome, anything has to do with STEC shedding. So remember, here's our STEC shedding profile of the entire population of animals, and these are the guys we really worry about here on the right, the guys that are shedding very high levels of the organism. And modeling this shedding is a whole other talk that I don't have time to get into, but trying to model that appropriately is not a simple task either. But keep in mind this distribution, knowing that we've got a few animals on here to the right that are shedding up to close to 1 million organisms per gram. We also have a lot of negative animals in the population. So now if we look at, we use a method called multidimensional scaling. So now we're looking at the functions that are present in the microbiome, and I'm superimposing on that, I've categorized the animals as to where they are on that shedding curve of 0157H7. And here now, I'm putting different things onto this multidimensional scaling. Sorry about that. So this is what the population looks like on the basis of the functions that are present in its microbiome, and you can see it sorts into about 2 to 4 different groups depending on how finely you slice the pie. If we look at the year or the cohort of animals, you see there's some association with cohort, but it does not fully explain it. If you look at gender, gender certainly doesn't fully explain it. I'd have been really scared if it did. Now the real question is, now do the data structures that we see inherent in the microbiome actually correspond with the STEC shedding? And the answer is, well, not really. At least not by this method. If you stand back and look at it and cock your head a bunch of different ways and you try and sort them into shedders just versus non-shedders or high, medium, and low, either way you really don't see any strong signal pulling that information. So that's by one method, a multidimensional scaling. If we come back now and take a different approach at it and say rather than trying to identify the data structures, let's see if we can use a machine learning approach that will actually take the data and learn from the data what the patterns look like and then put it onto a test set of data. So we've done that now. So we use Wilcoxon's rank sum test to rank the taxa based on their ability to discriminate just the high shedder and low shedders. All right, and we've set that cutoff at different levels and tried this. And here's the organisms over here and the p-values for that test. So now we take that data, we take that set of organisms, and we use it to train a classifier. This is a random forest classifier that we're using. And, sorry about that. And now we take that classifier, and we use it on accuracy on the training data. Obviously, it's going to be very accurate. And then we try to predict shedding on potential unknowns. All right, so data that the classifier has not seen. And when we do that, we get about 97% accuracy on 250 samples that we've done so far. So it looks like there may be some real potential here for using these classifiers, using these machine learning algorithms to learn what the pattern is from a very representative data set. Those are the two key things, right? And then be able to apply that to a set of unknown samples and accurately predict which category an animal might fall into. And I think that is very relevant and very appropriate to the type of diagnostic situation you would be talking about with BRD. Same type of thing. You don't really know who all the players are. You've got a new technology that lets you see players you've never even seen before. And you want to try and make the association. There's all kinds of variables that are confounding the associations. Why not use a machine learning approach to understand the patterns that are in the data and see if you can get some predictive output from that. So the second thing I want to cover quickly on the diagnostic side is asking the question, can we enhance this diagnostic microbiology that's being done in this project too? So the strategy that the guys out at Mark are using for identifying and enumerating these 0157H7s and these other STECs from the feces, I mean, it's like I said, it's the old traditional grind it, find it microbiology. So the sample, there's a liquid culture-based enrichment, or they may even go to just directly to immunomagnetic beads. There's selective and differential plating. They have to serotype the suspect colonies, and finally, PCR confirm. It's a pain in the butt. Everybody understands that. So the question is, can we just bypass that altogether, and can we just take these culture-based enrichments and use metagenome sequencing and read mapping or assembly to identify whether or not our target's in there? That's a whole new concept, as it's not like using a targeted PCR. This strategy lets you see all kinds of different things, and it doesn't limit you to just whatever you can see with your qPCR primers. So in order for us to be able to do this, we probably chose one of the most difficult organisms to possibly do this on because of the nature of the distribution of the virulence genes and how significantly the genome of this organism varies. And Mike talked a little bit about that earlier, and I don't have time to really tell you a lot about it. But basically what we have to be able to do is to identify the signature virulence genes, and we've got to know that they actually came from the same genome, because convergent evolution is rampant in these organisms. They'll inherit the same genes from very independent sources. And so you have to know that these genes are in the same genome, that it's actually of the 0157H7 lineage, and that it's actually a potential problem. I'm going to bypass that. And it's actually worse than that, because 0157H7 is just one of the 6 bad guys now. So it's got, or one of the 7 bad guys. It's got 6 ugly sisters that we also need to be able to identify. And we're just trying to do it on 0157H7. So what we've done, and I won't bore you too much with the details here, but we've done pairwise alignments between 0157 and other E. coli's, other strains in the database, 0157s against 0157s, and the key is to identify those regions in the genome, those segments in the genome that are specific to 0157 and all 0157s that we can muster up. And when you do that and you look at the distribution of the length, here's the length and bases of those segments, you find that most of them are really short segments. And if I go back to that big, nice, pretty colored diagram and we zoomed in, you would see that a lot of those segments where there's variation in the 0157 genome compared to other strains, there's also a lot of variation across 0157s. So those segments tend to be very, very small. And if we look at them, oftentimes there are situations where gene B and gene C are sitting here in the 0157 genome. If you look at that same position in another lineage, you find gene B sitting next to gene Y. And you have another lineage, gene X, and gene B sitting next to gene Y. And the same thing over here on the other side. And those are actually perfect diagnostic targets for the type of sequencing that we do, because all we have to do is to look for contigs whose ends, look for sequence reads whose ends map to these 2 genes that are unique to that lineage. So there's a perfect way to go about, or perfect situation to use for the type of sequencing that we have. So we did this initially. So we took whole genome sequences of 0157, other STECs, other E. coli strains. And if you got to do this in silico first, because if you can't do it in silico, forget even trying to do it with real data. So we tried it in silico first. So we simulated. We took these genomes and broke them up into 100 base pair. These are what our reads look like from the Illumina machine. So we simulate that so that it looks like it's real. And we put them at different levels. So we put them at different ratios one to another to try and make it look like a real sample as best we could. And then we use this read mapping strategy just at those diagnostic markers to say how well did we do. And of course on this data, the simulation, we did quite well. So here are the different strains that we used, their genomes, and this is the relative ratio that we put those in each of the 4 different tests that we did. And if you look across here, if you just look down here at the bottom, here's the ratios that we actually predicted using this sequence-based strategy. So we did quite good on simulated data. We were really excited, like, wow, this might really work. You could actually go from an enrichment culture to diagnostics within, whatever period of time you could get the sequencing done. So we got really excited about it, and then the real world hit. So then we went into the actual enrichment cultures, and just putting this in binary now. So down here on this scale, this is a CFU per gram that we know that came out of there by the hardcore microbiology. And I just put on a binary of whether we detected it or not by sequencing, by this assembly-based strategy with our markers. And we did a little bit better than chance, not a whole lot better than chance, and there's really no pattern here. It's not as if the samples that were low in abundance of the organisms are the ones that were the false negatives. That range is about as big as it was in our false positives. So we still got some more work to do. This is not an easy task to do, and there's still a lot left, but I think there's hope. So prospects for diagnosis by sequencing. Hopefully I've taken you on this myriad of things that we've learned along the way of our projects. They're not really the main focus of our projects, but they're take-homes, and that is, I think these machine learning approaches from metagenomic data are really going to be very facilitated for looking at these polymicrobial diseases. So you'll be able to understand and define patterns and use these classifiers to pick out and sort animals or humans, whatever the case might be, into predisposed or not predisposed, or diseased and not diseased populations. It's going to require a very large database of well-characterized animals, diseased and healthy. That's going to be absolutely critical to train these machine learning algorithms on. So that database has got to be collected very, very carefully. And data, like you saw with our data, it can be an outcome of studies that are looking at the fundamental biology and ecology of the system. So you're learning a lot along the way as you're creating these databases. Right? So that's a take-home from that. And secondly, these metagenome diagnostics, there may be hope. Doing it for a single organism is going to be tricky, and it's really going to depend on the biology of that organism. And as I mentioned, we probably picked the most difficult organism to possibly do this with. But if you can do it with that one, that means it'll be easier to do it with the others. It's also going to require that we know something about the genomic diversity and the population structures present in these organisms. We thought we understood enough about O157 genetic diversity that we pinpointed markers that were truly diagnostic, and indeed you saw they weren't quite as diagnostic as we thought. So clearly there's a lot to learn. Lastly, I just want to acknowledge, I've already mentioned the group out at USMark. My group at UNL that I work with, Steve Kochman, Atsuko Maruyama, and Jennifer Clark, are fantastic faculty colleagues across the data sciences that really keep me, because I float off at 30,000 feet if I'm not anchored, so they really keep me anchored. Rohit Sena did all the programming. So he's done all the programming you've seen in all these slides, as well as all the analysis, and Tai Nguyen has been the one that's built all of our databases. And I'll stop there. Thanks.